Welcome everybody again to another episode of the ideaXme show, the show that profiles the human beings behind the really big ideas that are shaping our world, inspiring future creators, and for all those that like really great stories. I'm Ira Pastor. I'm your health, aging, longevity ambassador with you today. So for the last several shows, we've been spending a lot of time on the different hierarchical levels of the architecture of life and aging. We've been talking about the genome and the microbiome and systems biology and a variety of other topics. However, we can't lose sight of the fact that while all of these integrated and biologic processes are important in the maintenance of our daily health and wellness, that when things go wrong, the trickle down is unfortunately a wide range of chronic degenerative diseases responsible for human suffering and death. And Alzheimer's disease is one of these conditions that, while it may not be responsible for as many deaths as, say, cancer or heart disease, it is a problem that is growing substantially in our rapidly aging population and is predicted to become an epidemic of major proportions, both direct and indirect cost to society in the coming decades. Alzheimer's is an irreversible progressive brain disorder that slowly destroys memory and thinking skills and eventually the ability to carry out simplest tasks. Estimates vary, but experts suggest that more than 5 million Americans age 65 or older may have dementia caused by Alzheimer's, with an estimated 18 million worldwide, and the global burden is projected to grow to 14 million in the US by 2060 and over 100 million globally. So this is truly an area of substantial unmet medical need. And today there's no cure for Alzheimer's disease. Available treatments are relatively symptomatic and remain palliative. And the predominant area of clinical research to date, which is focused on treating what has been thought as the primary underlying disease pathology, that is beta amyloid plaque formation, and hence the attempted reduction by either traditional drugs, scavenging by immunotherapies, or vaccination for amyloid protein, have really been using billions of dollars of research funding and yielded what many consider lackluster results to date in the clinic. So much so that many members of Big Pharma are actually even getting out of the Alzheimer's space completely. So it's not a very great situation. This has prompted a set of thought leaders to ask a bigger question. That is beta amyloid the right target, or is it and its related tau neurofibrillary tangles and damage downstream from more important biologic events that we should be paying attention to? Well, I'm excited to say that we have one of those thought leaders with us today. Professor Ruth Itzhaki is Emeritus Professor, Division of Neuroscience and Experimental Psychology at the University of Manchester. With a bachelor's, master's, and PhD in biophysics, Dr. Itzhaki has had a career full of a diverse range of research, including activities studying iron binding in plasma, the effects of ionizing radiation on natural and synthetic macromolecules, the study of chromatin structure, as well as the effects of irradiation and carcinogens on chromatin, as well as the effects of viruses and neurologic diseases. Most recently, she's been specifically studying Alzheimer's disease and the role of viruses acting with genetic factors in dementia. Her group has been investigating whether common viruses, which many elderly people harbor within their brain, may have a role in progressive Alzheimer's disease, and they've been discovering that viruses located within plaques may potentially be the causative of the production of the main components of plaques and tangles, namely beta amyloid and amyloid tau. They've done a wide range of experiments and published on antiviral agents that may be effective in the treatment to slow Alzheimer's progression, and also looking at future possibilities for prevention of disease with vaccination. Her virus work has won her Investigator Award from the Lancet, a Wellcome Trust Innovation Award, two Olympus Foundation Awards, Alzheimer's Research Forum Award, and the Manchester City Council Award. With all of that, Dr. Itzhaki, thank you so much for taking the time to come on the show today.
Well, thank you. I'm glad to be here. We're talking with you.
It's an honor. Really just wanted to start out giving you the floor basically to introduce yourself, tell us a little bit more about your background, how you got interested in science, how you got interested in biophysics, and ultimately sort of your journey to this fascinating area of neurobiology, Alzheimer's, that is clearly at the cutting edge of sort of the study of this disease.
Well, I started off in physics because I was very interested from a very young age in science, and physics seems to be the most exciting. But then I gradually thought that I was more interested in working out how living things work. And so I changed over to biophysics and I was lucky to get the only studentship available in the UK at the time to introduce me to biophysics, which was then, a long time ago, was then really more or less technique oriented and rather physiology oriented, which I wasn't that interested in. But I really went into molecular biology, I suppose one could call it, um, when I moved to Cambridge and started a project on trying to work out something about chromatin, or as it was called then, deoxyribonuclear protein, very much a mouthful. And as I think I put it in my brief CV, I've had to write quite violent changes of subject, not through choice, but because circumstances. And the next thing I went on to was looking at effects of carcinogens on macromolecules. And so eventually, after many years, I came to learn something about viruses, and I found them extremely fascinating. And somebody I collaborated with was very interested in the possibility of viruses being involved in certain neurodegenerative diseases, in particular Alzheimer's. And a paper appeared, this was in the 1980s, a paper appeared which suggested that there might be an involvement of herpes simplex virus type 1, which is known as the cold sore virus, because in herpes encephalitis, which is luckily a very rare but very serious disease affecting only about 2 or 3 people per million, the damage it causes is mainly situated in the same regions as those that are affected mostly in Alzheimer's disease. That was the main connection. There are a couple of others as well, which are less perhaps obvious. But that led me to an interest in the disease, which anyway I'd seen in family, a certain family member, which is very, very distressing, of course. And I decided that if herpes isn't a factor in this disease, first thing one should do is try to find out if the virus or viral DNA is present in brain of Alzheimer patients. It seemed an obvious thing to do. And I think nobody had looked at Alzheimer's disease before. There were a few studies on looking for the virus in ordinary, normal human brain, post-mortem, of course, but they were very inconclusive. Some found evidence and some did not. The main reason, almost certainly, being that they were all rather inaccurate, or rather insensitive, not inaccurate. Really, they couldn't detect anything below a level of about 1 viral particle per cell, which is really, on average, very high level. Now, we therefore decided to use polymerase chain reaction, PCR, which is hugely more sensitive. And we discovered using that, after taking enormous precautions to avoid any contamination, which is always, it bedeviled it at the time because it was always a worry, we found rather disappointingly that virus was present in the normal brain, elderly normal brain, as well as an Alzheimer brain, which was a quite disappointment because we hoped naively that it would be present in Alzheimer's brain but not in elderly normals. But that problem was sorted out a few years later. Should I go on, or
Absolutely, please.
Okay. Well, we had awful trouble getting that paper accepted. People everywhere said it's contamination, you've got this and that and the other, which wasn't the case. And we managed to convince people, or some people after a while, but there was still a lot of hostility to the idea that there could be anything like a virus or any type of microbe or pathogen in brain. But we pursued it with great difficulty, I should say, because of getting funding and getting papers published. But what we decided to do was, we thought, if there is a difference between the two groups, which means one group develops Alzheimer's and the other one doesn't, could it be a genetic factor that the group that one group has a certain genetic factor that the other one lacks, and that might predispose them to the disease, or could be the other way around, it might protect them. So we decided to look at the known genetic factor, or the known susceptibility factor, apolipoprotein E, to find out what allele of that, what type, form of that might be involved. It was known to be, as I say, a susceptibility factor. That means that it wasn't didn't necessarily cause the disease, but it was probably involved along with something else, and the two in combination. In other words, on its own, it almost certainly wouldn't cause the disease. So what we found, we looked at the DNA that we'd already obtained from brain specimens, post-mortem brain, and we found that a very high proportion of the Alzheimer patients who had HSV1 in brain had an ApoE4 type 4 allele, whereas those who didn't have HSV1 in brain, or the controls, all had low values of ApoE4. So it looked as if there was a connection. And what was very intriguing was I had a sort of crazy idea, and I thought, I wonder whether cold sores comes into this, because cold sores is mainly caused by HSV1, although sometimes by the so-called genital herpes, HSV2. But most cases are caused, or were certainly then caused by HSV1. And so we looked, and to our great surprise, and really I have to say delight really, we found that a high proportion of people who had cold sores had an ApoE4 allele. So what it looked like was that damage could be caused in the peripheral nervous system, in the region where the virus lingers in the periphery. And I think it causes damage owing to the same factor, the ApoE4 type 4 allele, as in brain, which is rather a nice support, although indirect, for the idea that ApoE4 plus HSV1 in brain is a major factor in the disease. And we were able to show, not hugely convincingly, because our numbers were so small, we didn't have enough money to do more specimens, which of course we had to pay for. But it did look as if you couldn't attribute this to the fact that people with Alzheimer's were more susceptible to virus infection, or people with an ApoE4 allele were more susceptible to HSV1 infection, because this is sort of thing that's been argued since. Because the proportion of people with the virus in brain was fairly similar in Alzheimer's patients and in elderly normal. So we couldn't attribute the presence, couldn't attribute the disease to the susceptibility of the Alzheimer patients to infection with HSV1. And then we moved on. We decided to look for a possibility that antibodies to the virus were present in the cerebrospinal fluid, CSF. The idea being that it was based on the fact that in herpes simplex encephalitis, antibodies were found in the CSF for a long time after the episode. This is for those who survived what was then a very dangerous disease. And the antibodies persisted in it. So we thought if we look there, we might find evidence of active virus, not just virus as a passive sitting resident in the brain. And we looked, and to our surprise again, we found that around 50% of the people had antibodies to HSV1 in their CSF. The numbers were somewhat lower than the proportion with HSV1 in brain found by PCR, but we can explain that and say I won't do that now because it's published in the paper, but there is a perfectly reasonable explanation for that. So that showed that the virus was not only there, but it was also active. It was at least at some times replicating, although probably most of the time it was dormant in a so-called latent state where there's very limited transcription. Only the latency, so-called latency-associated transcript is made during latency. And as far as everybody knows, there's no protein, or if there is any, it's very, very little. So it might be doing damage. This is not to say that it isn't damaging in latency, but there's really no evidence one way or another to sort that out, or very little work has been done, mainly because it's difficult to get a model for it. So after that, I think the next thing we decided to do, or around about that time, was to look to see whether there are any obvious links between the virus and the disease. For example, we tried infecting cells in culture with the virus and looked to see if any amyloid was formed or any abnormal tau, which is the main component of neurofibrillary tangles. And amyloid, as I think you said, is the main component of amyloid plaques. And to our great surprise, and I have to say again tonight, we found, really surprisingly, that amyloid was formed. There was deposition of amyloid in these cells, shown by staining, and deposition of abnormal tau. So the two main features, the abnormalities of Alzheimer's disease brain, were reproduced to some extent, at least, in cells infected with the virus, in cells in culture. We also managed to get hold of some specimens of brain from mice which had been experimentally infected with the virus.
Mm-hmm.
And there again, we showed what looked like deposits of amyloid using staining by antibody detection. So this was quite encouraging. And because it looked as if the virus is capable of producing the main features of Alzheimer's disease.
Absolutely.
So we went on to look very, very tentative and a speculative study on whereabouts in brain the virus might be situated. And what we did was we used what was called in situ hybridization,
Okay.
where you don't extract DNA from the cells, look for viral sequences, but instead you look in sections of brain and find by staining them. The first thing we did was to find by PCR, which you can do in sections, not just on extracts.
Mm-hmm.
And it's very interesting and reveals types of cells that the virus is in. So that's useful, but very, very difficult technique. It took about 4 years to get it set up. And we stained also for the presence of amyloid. And what we found, again, to our enormous surprise, was that nearly all the DNA, viral DNA in Alzheimer's disease brain was located within amyloid plaques.
Mm.
And so that was really, and although I say the pictures were very pretty, this of course was attacked as always by the thought that this was an artifactual, and the plaques, everything stuck to them, they're sticky, and so you look for anything and you'll find it there. But we were able to discount that. I think we thought of about 6 or more objections which people might raise, and we refuted them all in various papers. So that was gratifying, and that did rouse quite a lot of interest, I have to say, but it was squashed again by the dominance of the antimicrobial people who for some reason couldn't contemplate the idea of any pathogen, infectious pathogen in brain. But the next thing we did, persevering in this, was to find whether or not in infected cell cultures, one could reduce the damaging effects, the production of amyloid and so on, and also cell death, of course, which is what viruses cause usually, if they don't go latent. Whether you could reduce that by use of standard antiviral agents. And we looked at quite a lot. In one paper, the main paper we published in, I think, 2010 or something like that, we found that acyclovir, which is the most used one, certainly it reduces the amount of amyloid that's produced, and it decreased the amount of abnormal tau almost to zero. So that was very good. And then we looked at a host of other antivirals which use different methods which act by different methods to stop virus replication. And we found in every case that there was a reduction, which was very gratifying. So I would say those are the main things that we've done over the years. And we faced a huge amount of hostility until about roughly 8 or 10 years ago, when other people entered the field, which they hadn't done before because they couldn't get grants, just in the way that my group and I couldn't get grants. But eventually some people did get some money, and they really confirmed and extended our observations, which was very good. And in the end, turned out, I think a few years ago, I counted, there must be now well over 200 papers, but I've lost count, which support either directly or indirectly our work. And they used very diverse methods for doing this, not just what we've done, but all sorts of other ones, GWAS, genome-wide association studies, epidemiological studies, and I think of virological studies, biochemical ones, and so on. And nearly all but 3 papers, about which we're not sure why they got different results, but all the others really confirmed and extended our work. And then the main, I think, breakthrough came a couple of years ago when the Harvard group found out that beta amyloid seems at least to act initially as an antimicrobial peptide, which was something we suggested way back in 2007 in a paper, but we weren't able to show it had antiviral properties. We did look, but probably because there were side effects. The amyloid that we used was a commercial product, probably not very good, and it or damaged the cells to the extent we couldn't really say for sure that it did have antiviral action. But anyway, and in fact, in 2002, another group in Australia who'd been struggling with the same sort of idea thought that probably the amyloid acted by encaging the viral DNA or the virus. And this came up much more clearly because that was only hypothetical speculation. Nobody took any notice of it, of course. But when the Harvard group found out that this was the case, that this antimicrobial peptide, they speculated that it works, amyloid acts by encasing or entombing pathogens. This was a very good vindication of what we thought and what somebody else had actually speculated about theoretically.
Absolutely.
So that was quite a breakthrough, a new attitude, which of course didn't go down very well. But then even greater one came in the last year as well, when the group Reedhead et al., Ben Reedhead and colleagues, and what's his name? Well, anyway, it doesn't matter. It's Reedhead et al. Paper in 2018 showed that not only was HSV1 present in brain, but also human herpes virus type 6 and 7. And that these were causing effects. They looked at networks and they found quite strong evidence that these viruses were causing an effect on various pathways of the cell and really causing damage. They totally implicated these viruses in changes in Alzheimer's disease, but without being able to prove it's causal. And I should say here that all the work that's been done, all of it showed an association, just as ours did, but there was no real proof that there was a causal effect. I mentioned the work that we did, which was published in The Lancet, which suggested that, but the numbers were too small, as I said.
Sure.
But then the papers came out from Taiwan, which very good evidence, although there were a number of flaws in the studies, but good evidence that HSV1 really was involved and probably was a cause, mainly because antivirals seem to very much reduce the frequency or the prevalence of Alzheimer's disease. There's quite complicated explanations why that might be. I published this in a paper with Richard Latham, Edinburgh, and we are hoping to repeat the work, I should say, because I think it needs repeating with various precautions taken that I have to say weren't obviously done. Perhaps they weren't in the position to do them by the people in Taiwan. But that really, I think, was the first evidence of a causal role of HSV1 as opposed to an association, because of course association doesn't prove causality. And probably the only way you can prove an agent is causal is either by preventing it by vaccination or some pre-treatment of some sort, which is specific for that agent, or by reducing its effect in people who already suffer so that their disease is lessened, really. So one or other of those procedures are probably the only cause for a disease which affects humans and which you can't therefore look at effects of injecting. Obviously, you can't inject the agent in humans for ethical considerations. So they're probably the two only methods, or certainly the main methods, for implicating an agent in a disease. Sorry, that was very long-winded.
No. It was a wonderful overview, and I appreciate you going into that detail. As you were just finishing up with, you remind me, when we talk about sort of the challenges in biomedical research of going against a doctrine and some ingrained doctrine. It brings to mind the work of Barry Marshall and H. pylori and peptic ulcers, and the challenging decades of medical doctrine. No, ulcers are not caused by spicy food or too much acid or stress, but there's this bacteria, and got him the Nobel Prize. And it seems that you're it's all on the same path. Can you just tell us some of the experiences, what you've run into in terms of this going against the, this major beta amyloid wave, that's all there is, some of the challenges, the surprises, are there colleagues of yours that, that they'll criticize you in the medical literature, but behind the scenes, they agree with everything you're doing. What are you running into in terms of this aspect of the biomedical scene?
Well, it looks like it. I mean, one problem was that they never produced any scientific arguments against it. This was our main complaint, that behind the scenes, well, I don't know what their thoughts were, but they were certainly damning our grant applications and blocking our publications. And the irony is, though, that we're not saying that amyloid isn't important. We think it is important, but not important in the way that they said or believed that it's the cause. I mean, they never suggested why or how it was a cause, in what way it was produced, let's say, more in Alzheimer patients than in others. So it was all very hazy, that side of it, and yet it was looked upon as the main point. And it is probably a major feature. We think it has probably very damaging effects after some reason it changes from being protective to harmful, and it's not at all clear why or how that happens. But that being the case, it gives it quite a prominent role, but just not a causative role. Absolutely. So, but the main problem, as I said, was they didn't ever say what they objected to in the both for the work that my group did on viruses or for the specific bacteria that have been implicated in it as well. I mean, the people who pioneered that work never really had any decent objections to it. I think in some cases it was a bit more problematic because some people didn't get the same results. There were probably somewhat fewer papers supporting them. It doesn't mean to say they're wrong. There might be all sorts of reasons, but they weren't criticized for any real theoretical objection or even experimental objection.
Obviously, you have you're where you are with your work, and I think about the billions. I've lost track of the billions that have been spent on, we'll call, other approaches that have not worked out too well. And the fact that some of the mightiest of the mighty of the big pharma space, Pfizer comes to mind, recently just abandoned ship and have gotten out of this. If in the fantasy world where $50 billion or whatever falls into your lap, and you say, Dr. Itzhaki, if we're going to go with your research, we want you to craft the future of sort of the clinical development of this thing. Obviously, obviously this is a fantasy situation, but what type of Alzheimer's pharmacotherapeutic industry do you vision? Is it going to be based on small molecules? Would it be based on vaccines? A combination of these tools? Because I see right on the precipice of, the translation of something that potentially, as I said, it could potentially stop this amazing, as they refer to it, this tsunami that's coming, which everyone knows is coming, and the existing approaches have not put a dent in it. What would you create looking out the next 10, 20 years to sort of the human translation of some of these ideas?
I would like very much to have had a clinical trial set up in which I would obviously, as a non-clinician, be an advisor rather than a partner.
Sure.
But we've tried over a period of 10 years and we've been repeatedly refused, again for no solid scientific reason. I mean, in one case, we were refused by the board, although at least 2 of the 5 reviewers had given us very high marks, and the other 3 marks were pretty good. So the average was 80-something%, which is quite high. But the board said 2 things. Firstly, that one of the clinical steps that was being taken was not the best one, and yet it was quite ironic that one of the co-applicants was the main Alzheimer's disease advisor to the UK government. He was a co-applicant. So to say that there was something clinically wrong seems very unlikely with the advisor. And the other thing was that we hadn't done any animal experiments. Well, we had applied to that same funding body 3 years earlier for funds to do animal experiments, and they turned us down with a very, very bad comment, very critical comment about the work. So, and yet here they were saying we hadn't done it. Well, we hadn't done it because partly because they hadn't given us any money. So the whole thing was bedeviled by that. But anyway, getting on to more positive things, one of the main things we want to do is try to repeat the epidemiological work by looking at data banks which exist in Britain and various other countries. They didn't exist when I first started the work, which was in about 1989. There was nothing there. And I thought, once we started getting these results, I thought, well, we must look and see whether we can get any evidence of people who have been treated with antivirals, how they fared. Couldn't get any data at all, but it's all been collected, I think, in about the last 10 or 15 years. And that was when I'm talking about, it's about 30 years ago now. So that repeating the epidemiological studies in Taiwan, obviously a very high priority. I also want to find out something about whether herpes acts on its own or whether it acts in conjunction with other agents like the other bacteria, or whether each works separately or any two work together, or what. I think this is very important from the point of view of treatment, as well as being very interesting theoretically. So I would say those are the main things that we want to do. And we're hoping to get funding. We'd also like to set up a clinical trial, because there is one which has started in the States, which I'm in a way very pleased about, but on the other hand, very sorry that we couldn't have set it up in the UK, where after all the work started, really. And without wanting to boast, that's where it all started. But we never got the funding, say, over a 10-year period of trying. But if we did get money, we would probably try an antiviral like acyclovir, or the biodrug, which is called valacyclovir, which is better absorbed, in combination with an anti-inflammatory agent, because inflammation is obviously a very bad effect of, very bad happening in Alzheimer's disease. So I think we would differ in that way from the New York trial. And also the results of that won't be out for about 3 years, so it's going to be a long wait for all of us to find out whether they've had any success. If they don't succeed, I think it's not because the theory is wrong, but possibly because they didn't use an anti-inflammatory, or maybe there are other factors involved that we don't know about. I mean, that's always the problem in science, that you can only know what you found out or what others have found out. You can't guess what other factors might be involved.
Definitely, but I think, you're pointing out how many paths sort of people have talked about, viruses, neuroinflammation, maybe even, as you were saying, some positive, let's say, aspects of the virome that, as we know, there's not just bad viruses, there may be good viruses that are involved in some immunomodulation. So a lot of things to follow, a lot of things to get done, and I'm just glad you're doing it and thinking about it. And if we can help increase the awareness about you and your results, I think it's important. Moving, on the show, we usually move from the science into a little bit into the science fiction as we come to our wrap up, and we usually pose the questions to our guests. Obviously, throughout your career, you've met a lot of wonderful scientists along the way, and mentored some and learned from others. I'm going to pose the question, though, about people that you might have wanted to meet but are not around anymore, that laid the foundation for some of this. They can be scientists. They don't have to be scientists. They can be philosophers or artists or celebrities. For Dr. Ruth Itzhaki, if you had the ability to go in my hypothetical time machine over here and meet somebody and talk to them for a little, who would that person have been for you?
Yes, well, I can think in music who I would like to have met, but
Okay.
that's not a different subject. Well, I would love to have met J.S. Bach, because I love his music, but that's not a direct consequence or related, obviously. As far as people who are scientific, I think I would like to have met Marie Curie, because when I was very young, I always admired enormously what she'd done. And for a woman in those days to do anything, I was just amazed, that she managed to carry on. Other than that, no, I can't think of anybody particularly who. I think it would have been good to have met Alzheimer himself, Dr. Alois Alzheimer, because he was the first to actually define the disease.
Those were
I
good
thought
answers,
email
and I'm
them
sure
to you.
Dr. Alzheimer's probably would have been happy to meet you to see how you're taking this all forward. So
I hope so. I hope
I think
so.
it's a very good answer. Well, I just really wanted to, once again, thank you. Obviously, you have a very busy schedule and involved in so much. Really thanking you for taking time out of your schedule to join us, talk a little bit about your journey, talk about your research and where you're going with it. It's extremely exciting. It's clearly, in this, as we said at the beginning, the unmet medical need that this disease is placing in front of us, and what's coming in the future. We need the approaches and the forward-thinking visionaries like yourself to suggest other paths. So this is extremely exciting. Thank you for the work you're doing for everybody. That's unfortunately, as we all get older, we have to, we might be bumping into this pathology. And so really, thank you for being there. Thank you for staying the course. And for everybody listening, once again, it's been Professor Ruth Itzhaki, Emeritus Professor of the Division of Neuroscience and Experimental Psychology at the University of Manchester. It's been quite eye-opening, and thank you for the time and taking to teach us and tell us a little bit about what you're doing.
Well, thank you very much for having me. I enjoyed talking with you.
It was a great pleasure.